Hello, everyone, and welcome to today's webinar, Unlocking the Genetic Code, Harnessing the Power of Human Genotyping Arrays for Accurate Polygenic Risk Score Analysis. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. I would now like to welcome our speakers, Dr. Anu Mittal, Senior Staff Scientist, Bioinformatics, Thermo Fisher Scientific, Dr. Paul Lacaze, Head of Genomics at the Victorian Heart Institute, Monash University, Dr. Giordano, Bottà, CEO and co-founder of Allelica, and moderating our panel today, we have Shantanu Kaushikkar, Director of Microarray Business Development at Thermo Fisher Scientific. Our first speaker is Dr. Anu Mittal. Anu Mittal will be presenting on the topic of microarrays for cardiovascular disease testing and the valuable insights PRS provides for these complex diseases. Dr. Mittal, welcome. You may now begin your presentation.
Thank you, Christy, for the introduction. Welcome to today's webinar on microarray technology in predictive genomics, where we'll explore how microarrays enable scalable and accurate polygenic risk scoring. Here's the agenda for today. We'll start with an introduction to polygenic risk scores and their role in predictive genomics. Then we'll discuss why microarrays are an ideal platform for PRS. Next, we'll dive into imputation-aware array designs and how it helps maximize genomic coverage for accurate PRS calculations. Finally, we'll wrap up by showcasing Axiom microarrays design capabilities and how they can be customized for specific PRS needs. So let's dive into polygenic risk scores. So most complex diseases like coronary artery disease, type 2 diabetes, and breast cancer are polygenic, meaning they are not caused by a single gene, but by the cumulative effects of many genetic variants. So PRS quantifies this overall genetic risk of developing a disease or trait for an individual by summing the risk alleles weighted by their effect sizes for hundreds to thousands of associated SNPs. So what's the real utility here? PRS allows us to stratify risk across a population, identifying those who may benefit from more proactive healthcare. For personalized genomics, it can guide prevention strategies, treatment options, and overall disease management by tailoring care to a person's genetic risk profiles. To achieve this, we need a few key things. Most PRS models are driven by common variants, but recent studies have shown that inclusion of both common and rare variants capture the full spectrum of the risk much better. So we need accurate genotypes for all associated variants to ensure reliable scores. And of course, we also need cost efficiency at scale for broad applications. Now let's look at why microarrays are an ideal platform for polygenic risk scoring. First, microarrays allow us to simultaneously interrogate hundreds of thousands of variants across the genome with high accuracy. This level of coverage is essential when you're calculating PRS for complex diseases because many variants with small effects need to be considered. Microarrays also offer high imputation accuracy across diverse populations. This is important because we need accurate genotypes, even for variants that aren't directly genotyped on the array, to ensure that our risk scores apply to different ancestry groups. When it comes to rare and difficult to genotype variants, which cannot be imputed well, microarrays offer highly accurate direct genotyping using analysis algorithms and probe design approaches. And this is crucial for capturing the full spectrum of disease risk. Figure on the right shows UK Biobank array, shows higher genotyping accuracy of low frequency markers compared to the low-pass next-gen sequencing. In addition to high accuracy, microarrays are highly customizable, allowing us to tailor the content to specific research or clinical needs. Another key advantage is scalability. Microarrays are cost-effective for genotyping large cohorts, making them ideal for biobank scale studies where budget and efficiency are important. Lastly, microarrays are a robust platform providing reproducible data quality, which is very critical for consistent and reliable polygenic risk scoring across different studies and populations. With that, let's take a look at how imputation-aware designs on Axiom microarrays maximizes genomic coverage. So imputation grid design starts with over 100 million SNPs and indels across the genome, all with verified performance in the assay. And the goal here is to select markers that will give us the most accurate imputation coverage across diverse populations. In panel A, you can see the distribution of these available SNPs across haplotype blocks. The haplotype blocks are represented as colored rectangles. These represent regions where genetic variants are inherited together. So panel B illustrates the approach of purely physical selection, where markers are chosen based on uniform spacing across the genome. The red markers here represent SNPs, which are selected for the array. So the problem here is that for some haplotype blocks, we end up tagging multiple times, while there are some haplotype blocks which are completely missed, leading to gaps in coverage. Now, panel C shows the power of an intelligent, imputation-aware approach to marker selection. By considering LD and allele frequencies across multiple populations, we can choose markers more strategically. Here, each haplotype block is tagged efficiently with minimum number of markers required for accurate imputation. This ensures that we have comprehensive coverage with fewer redundant markers, optimizing both the cost and performance of the array. So this imputation-aware approach is key for maximizing coverage and ensuring the accuracy of PRS calculations across diverse populations. Now, it's tempting to think that more SNPs mean better imputation, but that's not always true. Imputation accuracy is influenced by several factors, including haplotype block size, marker selection, algorithm, and the reference panel used for imputation. Adding more SNPs to an array can improve accuracy, but after a point, the returns diminish. The figure here shows imputation accuracy of an optimized array with 800,000 SNPs versus grids of varying sizes with unoptimized selection. As you can see, Axiom pangenomics array, which is shown in red, it's an optimized array with 800,000 SNPs and can impute with accuracy similar to that of a 2 million SNP array shown in yellow that isn't optimized for imputation. So a higher SNP density doesn't always lead to better imputation performance, but the key is smart imputation-aware design, which can maximize accuracy and efficiency. Next, let's dive into the design capabilities of Axiom microarrays. One of our strengths lies in the vast amount of verified content we've accumulated over time. This guarantees strong performance for diverse applications. We also use a proprietary algorithm for imputation grid design. This ensures that our arrays are optimized for diverse populations. Typically, we design arrays based on 1000 genomes project reference panel, but we also have the capability to use custom reference panels from sequencing initiatives. Even when dealing with large panels, our platform is built to efficiently handle the data. Another key advantage is the ability to fully customize our arrays. Along with the imputation grid, we can integrate additional content modules to tailor the array to specific needs, whether it's rare variants, disease-specific markers, or custom research markers. In summary, the flexibility, customization, and powerful algorithms behind Axiom microarrays allow us to deliver high-performance arrays, which are fine-tuned for the needs of different research goals and populations. As we wrap up, I want to leave you with a few key takeaways. First, polygenic risk scores offer a tremendous opportunity in personalized medicine, enabling risk stratification and informed treatment decisions across a range of complex diseases. We also discussed why Axiom microarrays are ideally suited for PRS. They are scalable, customizable, and provide cost-effective solutions for large-scale products without sacrificing accuracy. When combined with our imputation-aware design strategies, even arrays with moderate SNP density can deliver high imputation accuracy, which is crucial for research across diverse populations. With that, thank you for your time today.
Dr. Mittal, thank you for your presentation. Up next, we have an exciting panel with Dr. Paul Lacaze and Dr. Giordano Bottà, moderating by Shantanu Kaushikkar. They will discuss the challenges and solutions associated with microarray technology for population-scale testing, with a specific focus on the use of polygenic risk scores for critical health concerns like cardiovascular disease. Gentlemen, welcome. You may now begin.
Thank you, Christy, for that introduction. Paul and Giordano, thank you for being here today and being on the panel. I hope your day is going well. Early for some, a little later for others, but I think it's going to be an exciting discussion today. As we just heard from Anu about the Thermo Fisher's capability to design imputation-aware arrays, I thought today's discussion we can, focus on multiple topics around that. I want to kind of start out first by just talking about arrays and how they're used and why they're used, such as, arrays are, they can drive sample power, they're low cost, and they allow for, studies to be completed in a much more quickly, better ROI. Obviously, arrays are great for that purpose, but they can't be used for identifying rare variants. We can certainly genotype them. On the other hand, if you look at whole genome sequencing, right, you can actually discover those rare variants, and it's a great tool for that. Next-gen sequencing, though, it remains expensive for these large studies, and so you can't quite attain the sample power. So I thought I'll start the question with you, Paul, first. Can you offer your perspective on how the two technologies, microarrays and whole genome sequencing, are complementary, and how can they be deployed in a research program?
Sure. So I'm a researcher. I use all different genomic technologies in my research: targeted sequencing, whole exome sequencing, whole genome sequencing, SNP arrays. And I think it's very important to get the right tool for the right job based on what you want to do. And in the presentation, we heard about common disease, and sometimes the perception is that common disease is caused by common variants, but there's actually monogenic forms of common diseases like coronary artery disease and breast cancer that are very important and caused by rare variants. For example, in the LDLR gene for familial hypercholesterolemia, or the BRCA genes for breast cancer. And those rare variants need to be measured with the right tool, which is typically sequencing. Target sequencing can be used as a more cost-effective way to detect rare variants. And so I think that needs to be kept in mind when we're thinking about diseases like breast cancer and coronary artery disease. However, there's only a smaller relative number of monogenic variant carriers in the general population, whereas there's lots of people whose genetic risk we can stratify with the use of polygenic scores. I think we get into trouble when we use the wrong tool for the job and miss information that really is critical to the job that we're trying to do. And if that job is identifying high-risk individuals for earlier intervention and risk management, I think we need to prioritize the detection of the highest risk people first, and that would probably be the rare variant carriers, and then stratify the remainder of the population based on polygenic risk as a second priority. That's the way I see the different technologies being used in the context of kind of population scale genomics and genomic risk assessment and risk management.
So you're looking at basically whole exome for those carriers that have these monogenic, and then arrays for some, those that have more common variants, and being able to identify those that are at the highest risk. Combination of two technologies, really serving.
Well, yeah, here in Australia, we have a program called the DNA Screen Program, where we've designed a small targeted sequencing panel to detect rare monogenic variants in only 9 genes, for example, as a first line population screening tool. And then, so we think that's where it should start. And then in the future, we can add on different types potentially of other genes, or potentially polygenic risk, to further stratify the population. So that's the approach that we're taking. And it's a very low cost first tier screening tool.
Okay. Giordano, just a question. You've in this space and have a fantastic software and polygenic risk scores. How does Allelica integrate this rare variants for, around the monogenic variants basically into polygenic risk scores? Or can you give some examples of, where Allelica software is making an impact in bringing polygenic risk score closer to the clinic?
Mm-hmm. Yeah, absolutely. Thank you, Shantanu. And so there are 2 different pieces of information of our genetic diversity in all monogenic and polygenic components are independent, are orthogonal, and they both confer risk. So in fact, what we consider to be the next generation of clinical genomics is a clinical genomics that takes into account both component of our genetic risk. So they cannot be taken independently. So they cannot consider only monogenic or only polygenic. Shouldn't be one prioritized over the other one. They are both conferring risk. Rare mutations are extremely rare. If you think about that, for breast cancer, a recent paper in New England Journal of Medicine identified that just 2.6% of women in the population, they carry mutation, while with polygenic risk score, we can identify more individuals that have equivalent risk of monogenic mutations. But this does not exclude, the use of one technology or over the others. So they need to be considered jointly. And for monogenic mutation, it's important to use technology that can identify, any mutation. So the use of a targeted panel and microarray is the most effective and accurate way to assess this joint effect. Because polygenic risk score can identify, women at risk, for example, for breast cancer, but also can stratify the risk of carriers. Because if you think about the carriers of moderate penetrance genes, such as PALP2, CHEK2, ATM, they can be stratified and identify women that are below or above the guideline threshold. Even 50% women below, 50% women above the guideline threshold. So the knowledge to be just a carrier is not enough. So it's not accurate. We can do much better. We can do much better stratifying the risk in carriers. So these 2 pieces of information needs to be considered together. And advancement in a clinical setting, for sure, with breast cancer and also coronary artery disease. Because if you think about that, up to 30% of heart attack cases happen in individuals without traditional risk factors, so without high cholesterol, high blood pressures, are not smokers. And this is a big missed opportunity to prevent those heart attacks, because we know that heart attacks are highly preventable if we can identify individuals at high risk. So polygenic risk score can identify around 20% of a population with more than 2-fold increased risk. And these allow targeted intervention, to really allow the implementation of precision medicine for common disease. So these are 2 main applications that we are seeing, getting traction, and also followed by prostate and colorectal cancer.
Okay. I think there's a statement you made, Paul, which is, using the right technology for the right job, and certainly that resonates. I think that's what I would say. We talked a little bit about, we alluded to a little bit about costs, microarrays, whole genome sequencing, and how they're complementary. I want to kind of play that into the polygenic risk score software and ask Giordano. Giordano, what trends have you seen in genomic testing prices overall, and how has price impacted reporting solutions? If you can offer a comment on that.
Yeah, absolutely. So, cost is important because here we want to genotype more individuals compared to what you can usually do with diagnosis of rare conditions. So here we are implementing genetics and genomics in common diseases. So these diseases, by definition, are present in many more people compared to rare conditions. So having a solution that is cost effective allow also the implementation in clinical setting in a much faster way, because so the health economics at the end they can allow the implementation also from a reimbursement. It can be like from a public health system or insurance perspective that are very price sensitive. So for sure, these allow the implementation in clinical setting, but also in performing studies at population level that can be done in many more individuals compared to what you can do, for example, with the whole genome sequencing, that you can identify all variants present in a human genome, but then it limits the number of individuals. And we know that many of these common diseases, they're caused by a combination of both common and rare variations, but the common variations, they have a very small effect. So you need to have many individuals with that common variants to identify them. So having a tool that allow to scale the number of individuals that can be genotyped is really effective.
Okay. Thank you for that. I think we just heard the presentation about imputation-aware arrays, and the design capabilities to improve the performance of polygenic risk scores. Sort of continuing along the questions, Giordano, how do you integrate imputation panels? What are some of the imputation panels Allelica can integrate into their software? Public, custom, can you talk a little bit about that?
Yeah. So by default, we have a panel that harmonized two different reference panels. So the 1000 genome that comprises around 2,500 genomes sequenced at high coverage from diverse human populations, and also the human genome diversity panel, so the HGDP, that has around 4,100 individuals, also that from diverse populations. But also we can integrate custom panels. For example, if a research group of any user has interest in assessing the imputation of specific population, and they have even 50 whole genome sequencing individuals, this can be added to the panel to impute more variants.
Okay. And Paul, you talked about the screening. Sorry, you were saying something, Giordano?
Just so that when then we apply these reference panels to calculate polygenic risk score, we perform clinical validation in a clinical data set where we know the clinical phenotypes, so we can assess if the improvement of a certain number of genomes in the reference panel can allow improvement also prediction, or vice versa. If you use a certain panel in a certain population, does not add anything new. So it's very important to then align these new reference panels to a clinical distribution of individuals to assess the change in predictive performance.
Okay. Paul, you mentioned screening, that you're running a screening program today, and I'm sure there are other programs that have been run in the past as well. From an imputation perspective, can you offer some comments on what you've been using in Monash, and then also just in general imputation?
Yeah, so we have used those traditional reference panels that Giordano mentioned, but recently for the Esprit cohort, which is another cohort I work on, we've re-imputed the whole cohort again, about 15,000 samples using the TopMed imputation reference panels. And we think that's significantly improved imputation, especially in non-European individuals. And it was quite an undertaking, but we've almost finished that. And there's still some important limitations with imputation that people should know about. And one is that you can't impute rare variants very accurately, so you're still not able to capture that under 1% rare variant genetic variation, which are often the highest effect size variants. But also that what you end up with after imputation is about 90% imputed variance and about 10% direct measured variance from the array.
Yeah.
And sometimes when we're giving genetic risk information to people through a polygenic risk score, that means the majority of those variants have been imputed, which that individual may not know about, or the clinician maybe doesn't even know about.
Yeah.
And as we're moving to meta polygenic scores, like that use millions of variants, like all of the long tail underneath the genome-wide significance threshold, and that it does work better when we use millions of variants in the polygenic score. We basically have to impute. There's no getting around it. So I know that some projects are designing a custom array with just like the 313 genome-wide significance SNPs for breast cancer to do a direct measured polygenic score.
I see.
I think a lot of other groups are going to the meta GRS approach, which makes imputation, critical and important to get right. And imputation is going to get better and better over time as the reference panels continue to become bigger and more diverse. But that also means that you have to update pipelines, and the performance of the scores might change over time. So there's some important implications there with regards to delivering a test that gives the most comprehensive or best genetic risk assessment possible at that point in time, whereas in 10 years time from now, it might be a little bit different and hopefully improved, and that's important for clinicians and patients to understand as well.
Yeah, certainly we see infrastructure improving in terms of speed, and hopefully that we can use these larger imputation panels and make a difference. So Paul, just we've been talking about polygenic risk scores and for various clinical endpoints, and the ultimate goal is to identify those that are at highest risk. Obviously, we look at the monogenic, and those are the ones that are at highest risk. But there are those that have sort of you look at non-communicable diseases, and you're trying to identify those that may be at highest risk, and they have the common variants. When you look at polygenic risk score, what we're trying to do is sort of stratify them and identify them, much earlier than before a clinical diagnosis is made, either through like a MRI or CT or a blood test. From a clinical endpoint perspective, where do you think which of those should be tackled first, and that could make an impact to global health?
Yeah, that's a great question, and important that every clinical outcome has a different genetic architecture, and some are more polygenic, and also some just have a higher heritability than others, like they're more genetic than others. And so if there's a trait that's not particularly heritable or genetic, then genetic risk prediction is never going to work extremely well for that outcome. Whereas for others with a higher heritability, like coronary artery disease or breast cancer, we're going to be able to make a huge impact with genetic risk prediction for monogenic and polygenic. And the other important part is like how well the traditional or clinical risk assessment model already works. So for coronary artery disease, we're actually pretty good at predicting future risk based on traditional risk factors like cholesterol, smoking, age, sex. And when you add genetic risk assessment, it does improve risk prediction, but there's a debate about whether it improves it enough to be implemented, in primary care and primary prevention. And for other diseases where the risk prediction is not very good using traditional risk factors, or even doesn't exist at all, adding genomics might make a big difference there. So one example that we've studied is osteoarthritis and hip and knee replacements.
Yeah.
And they're traditionally pretty hard to predict, or there's no clinical risk prediction really happening. And we found that a polygenic risk score actually was associated with hip and knee replacements in the Esprit cohort of older people. And so, that might be an area where it starts a new risk prediction focus that hasn't happened before. So I think it's going to vary by disease outcome, and probably the diseases that have the highest heritability or genetic component will be the ones where polygenic and monogenic risk scores will be able to add the most value first, or ones that don't have any good prediction models yet.
Okay. Giordano, do you get any questions? What kind of questions do you get from your customers? You already talked about, breast cancer and coronary artery disease. Beyond that, do customers come to Allelica and say, do you have a specific polygenic risk score for a specific disease? Can you sort of give a little bit of insight into some of the others that you hear about?
Yeah, then there are a lot of interest also in trying to predict outcomes also during clinical trials to help in making clinical trials more accurate, also reducing the number of participants, because we can enrich trials for individuals that will have a certain outcome in a shorter time frame. So PRS can be used to reduce the number of participants and also accelerate the outcome of the trial. And this, has a very important outcome, and because if you think about that, the trials are very expensive and very long. If you can parallelize the trials with the same budget and try to get to the endpoint faster, we can have, in 10-year time, more drugs that can reach patients. So this can really have a tremendous impact in how we develop new medicines. And then also so we help our customers in developing new polygenic risk score for different phenotypes or traits that can be used in both clinical settings, also for research, because we have an engine that is called the Discover that implement more than 11 algorithms in parallel and then build meta-polygenic risk scores. This also helps to stabilize the signal, and we do that in multiple ancestries with also ancestry-specific distributions and ancestry-specific scores. So this can accelerate, the research and reduce trial and error that we often see when new research groups are trying to approach this field, and also often know that they can develop suboptimal polygenic risk score because of the complexity in developing, and so we are helping them in be sure that they can extract all the signals from their specific data set, and also that their score is validated in different ancestries and not based on self-reported, because also this one is very important to start an analysis with an ancestry analysis than relying on self-reported.
Yeah, I think the access to the clinical health records, right, is extremely important in the calculation of polygenic risk scores. And so really, it's more for a clinical setting than from a direct-to-consumer type setting.
I think this point on risk stratification being very important and a huge opportunity for polygenic scores, including like stratification for screening and prevention, but also stratification for clinical trials, as Giordano mentioned. And we, some studies I've been involved in, when a trial was not significant or found to be successful in the overall result, when we reanalyze the data, stratifying by polygenic risk, we can actually find a significant signal in the trial. So I think there's huge opportunities there into the future.
Yeah. And then, of course, the ultimate goal is to see the PRS being deployed in the clinic. Paul, you had a fantastic article, sort of an editorial as well, in Frontiers in Public Health in 2019 that mentioned, the current levels of public involvement need to be improved as the level of genomic research and testing in society approaches population scale. And we're certainly seeing that, happen. Can you expand a bit on the public involvement bit, and how can industry and academia collaborate in this area? How can the three groups represented here are exactly the kind of collaboration who should collaborate, I would say.
Yeah, no, I think that the public interest in genomics is just increasing, and the public awareness and the demand actually for testing is increasing. So we need to find ways to deliver genomic testing to more people in the best way possible. And I think that traditionally there's been a bit of a transaction where the research study or even the company has been benefiting more from that person giving their sample than the person has. So it's been a bit of a one-way street. And I think we need to turn that around where we're actually doing it to deliver direct benefit for the public and the population. And we're approaching that threshold, I think, where we know what information is going to be useful to people. We're trying to figure out how best to deliver it. And it's not about amassing, data sets anymore. I think it's about delivering benefits through genomic testing to the general public at a time when they're starting to, demand it more and ask for it more. And so we need to figure out the way to best do that. And we have to think about things like data security, data privacy, and informed consent. Like, what are they signing up for? What's going to happen with their DNA sample? What's going to happen with their data? And we need to get all of that right if we're going to really do this the best way at the population scale.
Yeah, I think it goes back to sort of return on investment and the investment that the participant is making in terms of its sample, and they want to see something back, right? And it's sort of upon us to make sure that they benefit from that, the discoveries that are being made. Giordano, do you sort of switching to, do you have sort of any comments on this, and how do you see your, Allelica sort of involved in sort of being involved in the public aspect of it, where you're explaining how the scores work, maybe, or how the software works? Can you talk a little bit about that and the public involvement piece?
Yeah. So the education is fundamental, because here we are really bringing something new in clinical genomics, because physicians or clinical geneticists are trained considering monogenic as the main cause or the main genetic component that causes diseases. Instead, there is the component of common variants that was not part of the training of the physicians that are practicing now. So it's really important to let them know the existence of these two different components that they both confer genetic risk of diseases. And then implementation in clinical setting can follow, because they can understand different implications, and also can understand the limitations of the current traditional risk models that are, for example, based on models that work well on people, for example, for cardiovascular, that are just older than 40 years of age. Instead, with the PRS, we can really intervene earlier when plaques, for example, that cause heart attacks have not been formed yet. So we can provide information earlier in life without waiting for the onset of traditional risk factors that often are too late. Because when we see, for example, is a high cholesterol, high blood pressure, and others, there are already plaques formed. Instead, here we can have like an early intervention, and we can really prevent diseases before of the subclinical onset. Because many diseases that are kind of discontinue from subclinical, and then they end up to have like a clinical manifestation. But we don't want to wait the clinical manifestation, because now we have tools to stratify the population and intervene early on. And then also the health economic studies, they show that is cost effective, cost savings also at scale in a public health perspective. So this is very encouraging.
Okay. As we sort of go into population scale, we talked a little bit about needing infrastructure, right, for the for imputation or for the reference genomes. Paul, a question to you. What sort of local infrastructure capabilities are required in the analysis of data for population scale? Can you talk a little bit about your experiences in Australia and elsewhere as well?
Yeah, I guess it depends on what you mean by population scale. If it, hundreds, thousands, tens of thousands, millions of samples, and which technology you use. So if you're doing whole genome sequencing, the requirements are, very, very much higher than if you're doing arrays. And the imputation also, it depends if you're using an external server or whether you're trying to do that locally. That can be quite computationally intensive. It's still, significant compute is required to do population genomics. There's no doubt about it, and that's going to be the case for a long time into the future. But certainly working with array data is a lot easier and less intensive than working with some of the large sequencing data sets.
And if, we will have a lot of customers listening to this call, what advice do you have sort of for the countries that are just starting out in this area, and what are the considerations for the research program to build local capacity? We hear, several countries in sort of the Far East as well as Africa, really, they want to benefit from all the discoveries that have happened. Can you talk a little bit about that?
Yeah, I mean, it's hard because genomics can be used for so many different things. There's so many possibilities and application areas, but I think you have to focus on the things with the best return on investment, and partition them into, cancer, rare disease, public health. They could be quite different. I do think it's important to remember where the world of genomics has come from, which is rare variants, rare disease, monogenic, and to then add this powerful new tool of polygenic risk scores on after or to follow that. All of the evidence and the knowledge we've developed in clinical genetics over the decades will still hold true into the future, and particularly for countries that are starting to develop genomic medicine infrastructure, and to keep that in mind before leaping into polygenic scores would be part of my advice.
Okay. And Giordano, just sort of looking at it from a software perspective, we've sort of, the early days saw UK Biobank data being generated from Axiom genotyping, and they've been, it has been used to generate several PRS scores. It's made a tremendous impact. Can you talk a little bit about, how countries can implement software and how they can bring genomics closer to the clinic, predictive genomics closer to the clinic?
Yeah, absolutely. For all new countries that they want to start genomics programs, really important that they collect highly accurate clinical phenotypes. So ideally they will need to combine the genomic information with electronic health records, so to also follow the patients over time, so individuals that that are part of the biobank in a prospective manner. So in this way you can collect clinical phenotypes and then apply all different models and algorithm. But the most important thing is to have a clean and data from a clinical perspective. And then apply these models at scale, increasing the number of participants and also follow them over time.
I think we're getting to the end, but I do have a question for you, Paul, which is, you completed the ASPE study a while ago. Can you talk a little bit about that and sort of what were the resulting conclusions and how did that study inform your research program and your screening programs that you're doing today in predictive genomics?
Sure. I'll try to cut a very long story short, but the ASPRE trial was a large randomized clinical trial for daily low dose aspirin in 19,000 older people. And it went for about 5 years and no overall benefit was seen in people taking aspirin versus placebo for disability-free survival or prevention of cardiovascular disease versus the bleeding risk. And fortunately, ASPRE set up a large biobank associated with the clinical trial, which enabled us to do genetic analyses. And then we could stratify and analyze the trial data alongside the genetic data. And the trial fortunately collected a diverse range of outcomes: deaths, disability, dementia, cancers, all different types of cancers, cardiovascular disease, stroke. And so and then, things like joint replacements and other phenotypes as well. So we have been able to now do, over 30 polygenic risk score studies from the cohort of all these different outcomes to assess and validate how the addition of a polygenic score improved a traditional risk prediction model in this excellently characterized prospective cohort of older people. So it's been extremely fruitful and diverse in terms of the genetics we've been able to do, but it's also kind of raised some factors to consider about when is the best time to do genetic risk assessment. Is it when people are older and maybe for common chronic polygenic disease, it would make sense, but then for other disease or for maybe even in general, maybe we should be doing genetic risk assessment as early as possible in adulthood to empower people with genomic risk information, like when they can really do something about it early in life. And then it can be used, for prevention, for primary prevention, risk stratification. So that kind of has led us into designing the DNA Screen project, which is specifically doing genomic screening in 18 to 40-year-olds in Australia. So kind of the other end of adulthood, to try and empower people with genomic risk information early and see how they can use it over time. And so far we're focused on these high-risk monogenic variants only, but we're thinking about how the future would look and what other genes or conditions or information might be useful and cost-effective to deliver to people in early adulthood through a public health screening program in Australia.
Yeah. I think one thing that struck me as you sort of went through this ASPE program, you talked earlier on about hip replacement and knee replacement. So then there's a certain segment of the population that could benefit from PRS scores. You want to avoid the resurgeries and so forth, and certainly stratifying that older population for these kinds of therapeutic interventions. What I think they could.
But yeah, what I did mention is that when the primary trial saw that there was no overall benefit for aspirin versus the bleeding risk. But when we re-stratified the cohort by polygenic risk for cardiovascular disease, we found that the top quintile of polygenic risk would have benefited from aspirin. And there was significant reduction in cardiovascular events if you stratify by the top quintile of polygenic risk. And the benefit then outweighs the harm of bleeding in that genetic high-risk subgroup.
I see.
So this is what I'm saying, that there's a huge opportunity for genetic risk stratification to maybe guide the use of a common drug like aspirin to actually maximize benefit and minimize harms in a subgroup of the population rather than a one-size-fits-all model. And I think, you know, the same might be true for statins and some of the other common drugs.
As we sort of close this, session today, any sort of last comments that you want the viewers to, hear from you? I'll start with you, Giordano, and then switch over to you, Paul.
Yeah, just know that now, it's clear that there are, two major components, of genetic risk that are caused by common and rare variants. Like considering one or just the other is an incomplete assessment of our genetic diversity. Then also methylation. So we have there are other components as well that then can be added over time. But now, what I think, is really important to consider is that, this continuum of the allele frequency, so the genetic variants that cause our diseases and, different traits, and they need to be both considered when we approach, clinical genomics.
Okay. Paul, any advice for our viewers today?
I would agree with that. And, unfortunately, there's been a bit of a chasm or a split in genomics with monogenic and polygenic. And it's because of the technologies that we have, which are both fantastic, but they're just different. And it's caused this different way of thinking. But really, they're all genetic variants in the end of the day. And it is a spectrum of frequency, and they're all important. And where we need to get to is comprehensive genetic risk assessment that doesn't have the one or the other, but has all of them. And there's a temptation to use SNP arrays and polygenic scores because it's so cheap or cost effective. But I think we have to resist that temptation and try and get towards the goal of the most comprehensive genomic risk assessment we can for a person to actually do the risk assessment as best as we can. And I feel like there's still work to be done there in how that is actually delivered. Because if you have to do 2 tests instead of 1, it's cumbersome and more expensive. Yeah. But, I think we have to think about the underlying biology and risk and clinical utility first, and then the right tool for the job in terms of the technology thereafter.
Yeah, I think you said it well. I would like to thank both of you for your time here today. I think it was fantastic and very useful. I think we have some work to do, definitely. I think there'll be some other seminars, and we'd love to have you back on the discussion panel again. Really appreciate it, and good luck in your screening program, Paul's, and the other future studies, and Giordano on the Allelica software and implementations.
Thank you.
Thank you.
Thank you.
Yeah.
Thank you, gentlemen. And for our last presentation, we invite Dr. Giordano Bottà to speak on how Allelica enables laboratories to become leaders in precision medicine. Dr. Bottà?
Hi, everyone. Glad to be here and to present the Allelica Predict software and its application in cardiovascular disease. Allelica enables laboratories to become leaders in precision medicine because we enable them to build a physician's-ready report starting from raw genetic data, and we automate the entire process to end up with the world-leading clinical reporting. This is a high-level description of the Predict workflow, where we start with a generation of genetic data, for example, from PMDA or any other microarray data. And then we align the genetic data with a polygenic risk score panel. To do that, we apply an imputation procedure to enrich the data set, so we can have millions of genetic variants starting from a microarray. Then we identify polygenic risk score percentiles in ancestry-specific risk distributions that allow us to finally predict risk of individuals based on their genetic data. But let me get into the details of how we can get to the final risk estimation. So we start drawing a distribution of raw polygenic risk score values, and then we can calculate the relative risk at each percentile. So we can identify thresholds that can allow us to identify individuals that are above a certain risk threshold. And these risk thresholds are very important because they align with the clinical guidelines. So we can identify individuals at 2- or 3-fold or more risk compared to the average of the population. Another very important aspect is that PREDICT performs multi-ancestry analysis, because if ancestry is not taken into consideration, the final risk estimation will be incorrectly identified, because each different ancestry has its own polygenic risk score distribution. So it's really important to have a multi-ancestry approach to the ancestry, to the risk estimation. And all the PREDICT analyses are based on models validated in diverse population. But our reports are labeled with this label, with allelic multi-ancestry PRS, because we identify ancestry-specific risk distribution, and then we assess the risk and then different PRS values, and also we calibrate the risk in each of the measured continental ancestries. Why is it so important to consider genetic ancestry and not only the ethnicity or the self-reported? Because there is a wide variation from what people consider to come from or to be, or which ethnicity they belong, to the actual genetic ancestry that identify individuals and group individuals based on the similarity of their genomes. But as you can see here, we can reclassify individuals based on their genetic ancestry compared to what is possible to do with self-report only. But let's dive into the application of predicting coronary artery disease. So why is it so important to estimate polygenic risk score for a heart attack? Because there is a big unmet need where up to 30% of heart attack cases happen in patients without standard risk factors. They don't have hypercholesterolemia, they have hypertension, diabetes, or they don't smoke. So they are invisible to traditional risk factors, and guidelines cannot identify, so models using the guidelines cannot identify those high-risk individuals. But thanks to the use of polygenic risk score, we can identify individuals that are at 3-fold increased risk compared to the rest, so the equivalent risk of those with a familiar hypercholesterolemia, even if they have average LDL. So these individuals, they go to do their annual checkup, for example, they see that they have average LDL, but their average LDL interacts with polygenic risk score, so with their genetics, to confer a much higher risk. So the two data points need to be jointly considered when we estimate risk of a heart attack. The output of PREDICT is very extensive and comprehensive, so it provides a PRS value, the polygenic risk score percentile, relative risk and lifetime risk, and also a genetic ancestry breakdown of the individual analyzed. And everything can be summarized in a PDF report that can be shared also with physicians and patients. PREDICT can be implemented in genetic laboratories, both on-premise and also in the cloud as a software as a service, and also are available APIs to streamline the process of to move data from different servers. So thank you for your attention, and I pass the ball back to the moderator. Thank you.
Thank you, Dr. Bottà. And thank you to Dr. Mittal, Dr. Lacaze, and to Shantanu Kaushikkar for their time today and for their important research. I'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for for underwriting today's educational webcast. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your friends and your colleagues who may have missed today's live event. Now, if you missed filling out a survey that's on the bottom left of your screen, we encourage you to do so now. Your participation is appreciated. Now, to all our friends out there, thank you for joining us. Have a great day, everyone. Bye-bye.